This is the first video of the Mouse Genetic Approaches for Neuroscience lecture. The objectives of this entire lecture are to know what are the different types of genetically modified mouse models, understand the benefits and limitations of different animal models, and also understand how to achieve targeted gene expression in mice, which is an essential tool in neuroscience. In this video, we will focus in describing what are genetically modified animal models, with emphasis in transgenic animals. Other animals or other genetically modified animals are knockout and knock-in animals that we'll also talk about, but in video number 2. So as for the definitions and classifications, there are 3 main classes of genetically modified mice. Transgenic mice are animals in which an extra gene is randomly added to the mouse genome. On the other side, knock-in animals are animals in which an extra gene is inserted into a specific locus in the mouse genome. And knockouts or KO or null animals are animals in which critical portions of a coding region of a gene are removed so that no functional protein is produced. And the same kind of principles can be applied to other organisms. So for this video, as I mentioned earlier, we will focus on talking about transgenic mice, what they are and how they can be generated. For generating transgenic mice, we need to start synthesizing or generating the transgene that we want to introduce in the animals. Transgenes are usually about 100 kilobases or less, and they're developed from plasmids and include a promoter region, which may be tissue specific, and a coding sequence that could be the cDNA of a gene of interest. The transgene may also contain several coding genes, such as this case, in which we have our gene of interest, an additional gene that can be used as a reporter. The foreign DNA is inserted into a fertilized oocyte or cells from the early embryo to transmit the foreign DNA stably in its germline. The most important characteristic of transgenic animals is that the transgenes are incorporated randomly into the genome and are influenced by the position at which they integrate. We cannot control where the transgenes go or how many copies are inserted. So there are different ways of creating transgenic mice, and in the next few slides we will see 2 examples of how to generate transgenic mice. This particular case describes pronuclear microinjection as a way to generate transgenic mice. For this, we will need a fertilized oocyte in which the nuclei from male and female have not been integrated yet. We will use a microinjection pipette and inject the transgene into one of the nuclei. What we will use is a linearized transgene, cDNA transgenic, and what we want to happen is that this linearized construct gets inserted in the genome by using nicks or breaks that occur naturally in the genome. Unfortunately, very often what happens during this process is that several copies of the transgene get inserted into the same place, like in this fashion, head to tail copies. And they also get inserted into multiple regions in the genome, which significantly enhances the expression of the transgene. It may not be physiologically relevant in some cases. Once the injection has occurred, we let the nuclei to integrate and fuse. And then once this is happening, we follow up with implantation of these fertilized oocytes into pseudo-pregnant females. After that, the offspring that these females produce will contain between 10 to 30% of transgenic animals. We will have to screen all the obtained offspring and select those that got the transgene inserted. And then from those, we will also have to check which ones have the gene integrated into one place or different places, where the expression levels of the different animals, and select those that more accordingly go into what we want. So this is another example for creating transgenic mice that uses DNA transfer into stem cells. Overall, for this technique, stem cells that are obtained from the blastocyst of one mouse line, in this case line A129, which has an agouti color, are transfected with a particular transgene. Then the stem cells transfected are injected into a host embryo, and they mix with the host cells during embryogenesis. This is implanted into a pseudo-pregnant female, and the offspring will be called chimeras, which is the result of different expression and patchy expression of cells from the host embryo and the stem cells transfected. The animals used are genetically marked with coat color genes, so that the contribution of the stem cells to the host animal can be determined easily by gene segregation and color distribution. So the animal that we choose as a pseudo-pregnant female has to have a different color. In this case is a C57BL, which is a recessive non-agouti and has this genotype. So the generated chimeras are crossbreed with an animal sharing the background of the host embryo. And only if the stem cells that were transfected and transgenic make the germ cells in the chimera, then the resulting animals from this crossbreeding will generate animals that have the color of the donor animal, in this case agouti color. And all the agouti animals are supposed to be transgenic, and they will be screened for the presence of the transgene, and we will remove all the black animals from the analysis. Then the resulting heterozygous mice are intercrossed to obtain homozygous mutant mice, usually at a 25% frequency if the mutation is not detrimental to the embryo survival and obviously development. And this technology has become the most popular method of generating transgenic mice, since it has a higher rate for generation than the previous method that we just described in the previous slide. So this is all about transgenic animals. Here I leave you with a problem that we will see in class that asks you to describe step by step how you will generate a transgenic mouse model of ataxia, expressing human ataxin 1 with 82 polyglutamines, driven by the CMB promoter and using stem cells transfected with the transgene. For that, then you will have to design the transgene with all the elements, and it'll be useful if you want to go back to the first video for the second lecture when we describe the technique, the DNA recombinant technology. And then we will ask you also to describe how will you transfer these stem cells and what is the technology that you will use. So we will do this problem in class, but I really encourage you to try to also do it in home, like at home after seeing this video.